So, hello everyone. I hope your week has been good. And in today's session, I'll be discussing the content of week 4. So, this will be the last session that we'll be meeting. So, if you guys have any queries about any content which were taught in previous weeks as well, you guys can ask me. Okay. And if you have queries, please feel free to discuss with me. So I'm Chandra. So let's begin. So I hope the screen is visible to you and I'm also clearly audible to you all. So this is the first question. The question here states, while advances in genomic methods have helped us to identify the genes mutated in genetic disorders, the challenge has been to dissect the mechanism by which the affected gene causes the disease. So which one of the following options offer an explanation for this challenge? So current advances genomic methods like the conventional methods, they have helped us, right, to identify the genes which have undergone mutation and thus causes a particular genetic disorder. But people have come across various challenges while doing this. So you have to identify which are these challenges that people have come across in this particular question. So the first option is the physiological function of most of the genes are unknown. Okay, so this is true. There are around 20-25,000 genes, and many genes we don't know how the translated protein actually functions in the cell, right? People are still studying about them. Then most of the genes they remain uncharacterized. This is also true. There are not enough biochemical assays to evaluate the function of all genes. So there are few powerful assays, but those assays are not applicable for all for evaluating the function of all genes. For example, for mental retardation. Okay, so it is quite difficult to develop biochemical assays which can help in identifying the defective genes which actually cause such kind of phenotype like mental disorder and all of these. So if you if you are in the meeting currently, so what do you think is the correct answer for this particular question?
Can you repeat the question?
Yeah, sure. So here the question states that while identifying the genes which have like undergone mutation, right? And these mutations generally lead to genetic disorders. So for identifying these kind of mutated genes, people have come across various kind of challenges, right? So and the options given here, you have to identify which are those challenges.
Can you state the options?
Yeah, sure. So the first option says the physiological function of most of the genes are unknown. Okay. And the second option is most of the human genes they remain uncharacterized. And the third, there are not enough biochemical assays to evaluate the function of all genes. And the last one is all of these. So these are the four challenges or defining some of the possible challenges which people have come across while identifying the mutated genes that are actually involved in a genetic disorder. So, what do you think will be the answer then?
I think all of these.
Yes, you're correct. Thank you, Heel. So, the correct answer is all of these. So, all of these options given above are actually the challenges that people have come across. Now, I would like to discuss in brief what do you mean by polygenic and monogenic disorders. Okay. So the term itself says, like from the term we can know that polygenic disorders generally involve multiple genes, right? Where monogenic disorders, a single gene is involved, correct? And why disorder? Because people have seen there's mutations, right, in single gene, which has manifests itself as a disorder, whereas in polygenic disorders, the mutations people have found in multiple genes, right? 3, 4, 5 genes, and there is an additive effect of all these mutations, so people get a particular disorder. And these polygenic disorders give rise to what is called complex disorders, right? Stop me if I'm not making sense to you, okay? And monogenic disorders, you are aware, right? So there are various disorders where it's like SOX10 gene, right, which results into a particular disorder, imprinted disorders, where only one gene is having deletions, right? So because of which a particular phenotype, disease phenotype, have emerged. And what about how these polygenic or monogenic disorders are studied? So people use positional cloning approach, right, to identify the unknown gene, right, which is resulting into disorder. And this unknown gene is single gene only, which is resulting into disorder. So they use a position cloning approach. Whereas for polygenic disorders, people use association studies, where association study involves there is a case group and is a control group. If you're aware, like from the lectures of week 4, so the case group have the individuals who are showing the phenotypes of a particular complex disorder, but the control group, they don't have that complex disorder phenotypes, and rest all phenotypes are same, like the age, they all belong from the same ethnic population, okay, etc. So people use association studies. Now, to identify this unknown mutated gene in monogenic disorders, people use microsatellites, satellites, which are nothing but repeats, right? So these number of repeats they find at particular locus within the chromosome, and they then track how these microsatellites are inherited within a family. And where the members of that particular family are having that particular monogenic disorder. In association studies, people track SNPs, okay, the variations. Now, these variations on their own, uh, like doesn't necessarily cause any particular disease phenotype. Okay? They maybe like the person can have if for the disease to happen, say there are 4 variations, 4 SNPs needed to be present in that particular individual, whereas that individual only has 1 SNP and rest all 4 are absent, so that person is living normally, right? And the expression of these variations or occurrence of this complex disorder also depends on the environmental factors, right? Like common cold. So in a class, there is a virus going on, viral fever going on. Few students get it, few students don't get it. So few students have the variations within their genome, which makes them more susceptible to that viral fever, where others they're not susceptible to it. So until they are exposed to that viral fever environment, we won't see this kind of segregation of the class. In normal condition, when there is no viral going on, all the students are normal, none of them have any cold or anything. So environmental factors also play a very important role in display of this complex disorder or polygenic disorders. So can you think of any other point of differences um, for polygenic and monogenic disorders? So let me ask you, do these two types of disorders show differences in how they inherit from one generation to the another generation? Is there any difference in the pattern of inheritance for polygenic or monogenic disorders? Any one of you can answer. No issues at all. So polygenic disorders, monogenic disorders, they they are inherited just the same way like we studied for Mendelian inheritance, right? Then both disorders can affect both sexes, both male and female, right? So in these aspects, these disorders are extremely similar to one another. The only differences are the one that I've listed down in this slide. So we have now recalled the concept of polygenic and monogenic disorder. Using this concept, let us try to solve this particular question, which is displayed on your screen. So which one of the following statements distinguishes a monogenic disorder from a polygenic disorder? Option 1, DNA sequence variation contributes to disease onset. Option 2, can affect either sex. Option 3, the affected individual can transmit the disease causing allele to the next generation. And the last option is none of these. So which of these option actually distinguishes monogenic disorder from a polygenic disorder? Can any one of you answer? Anyone? Karan, if you have just joined the meeting, right? Okay, he just left. No worries. What about others? Would you like to try? So I have tried to make this discussion a discussion where I request participation from my audience, right? So
I think option C, ma'am.
option C. So the fact you're saying that monogenic and polygenic disorders, they are different in the way they are transmitted from one generation to the next generation, correct? So I would say that you're wrong. Okay, so monogenic and polygenic disorders do not show any difference in how they transmit. Okay, they follow the same Mendelian inheritance pattern where the segregation happens, right? Homologous chromosomes segregate into gametes and all, and then they are passed on to the next generation by the mother and father. So that kind of pattern is same. Okay, and it can also affect both male and female. It doesn't show any difference in that. So the answer for this question is none of these. So none of these options actually distinguishes the monogenic disorder from the polygenic disorder. All three options are the characteristics of both the disorders. Do I make sense? Think over it
Yes, ma'am.
if you
Can you
have it.
please explain about positional cloning again?
So I was actually planning to have complete comprehensive discussion on positional cloning later on. But briefly, let me tell you that positional cloning is an approach that people have adopted to identify the unknown gene, okay, unknown single gene that causes a disorder. A single gene can cause a monogenic disorder, and this identification of this particular gene depends on we actually track the position of that gene in a chromosome. We do not rely on any functional assays. Functional assays means what is the function associated to this gene. These kind of studies, they are done after the gene has been identified, whereas the conventional methods are there which actually relies on the functional assays. Some people know that there is a biochemical process going on in the body, okay, and using the proteins that are involved in the biochemical reaction, they've identified the gene. That is a conventional approach. Or people use animal models where they do mutation of a particular gene of some candidate genes, and see the effect. So we are not using these approaches. We are trying to identify the unknown gene solely based on where it is located within a chromosome. Okay, so I will discuss more in detail in my next slides. For now, please have this up till now. I think should be clear to you.
Okay, ma'am. Thank you.
Yeah, great. So this was the second question. Now third question. So in this question, let's read two distinct genes that are likely to be inherited together. Okay, are called what? So there are two separate genes that are present at two different locus or loci, or to be precise, loci within the chromosome. Okay, and these two distinct genes, they will be inherited together. So what are these two genes called? First option is alleles. Another option is polygenes. Third option is linked genes, and fourth is none of these. This is a simple definition of, I would not say, I would request you guys to think over it and answer. This statement defines what kind of
Alleles.
genes. Okay, so you have answered alleles. So you're actually wrong. Okay, the correct answer is linked genes. Why? Let me explain. So say this is a, sorry, let me go back. So this is a homologous chromosome. So these two chromosomes are similar, not identical. Okay, and one chromosome comes from mother. So this has come from mother. Another has come from the father. Okay, so alleles refer to a mutant version of a particular gene. So if this is A and this is the locus, this box is representing the locus, a position of gene A in the mother homologous chromosome, maternal homologous chromosome. And from the father, a slightly different version of A has been inherited. Let's say it is represented as small a. Now, capital A and small a, they are present at the same locus. Okay, this is the locus. Now, these A, capital A and small a, these are the alleles. Whereas linked genes what are linked genes? They are present on different locus. This is locus 1, this is locus 2, and this is, say, another gene B is there. Now the position of A and B is such that they cannot be separated from one another during the recombination event. If you recall, when recombination happens, it happens during meiosis, right? Crossing over, it is also called crossing over, CO. Okay, so they are very closely positioned at a very close distance to one another, and people have seen the linked genes, they always inherit together because of this unique characteristic that they are positioned very close to one another. So when this individual, whoever having these two pair of homologous chromosome, like creates gametes of his or her own, so during recombination, AB will be, say at this position, recombination happens. So it will happen like this. AB will be inherited together into the one of the gametes. So this is a unique property of linked genes. Am I clear here? Why alleles will be not the correct answer? Rest of you, am I clear about what are linked genes and why alleles is not the correct answer to this particular question?
Yes, ma'am, clear.
Okay, thank you. Now,
Ma'am, I have a doubt whether if allele, there are two alleles means only one allele will be passed down inherited or both the alleles together. Like how the allele is inherited.
okay, Sure. So suppose this homologous chromosome is present uh, in an individual who is a female. Okay. And now this individual has mated with the man. Okay. And this person has his own pairs of homologous chromosomes. Okay. And at this particular locus, one, uh, there is, say, another A and another allele, say A prime. Okay, so the distribution of these homologous chromosomes, it follows the law of segregation, right, from the Mendelian inheritance, where it says that these two homologous chromosomes, while gamete formation, they will have to separate. And while separating, the genes which are present, that is the alleles which are present in that particular locus, they will also separate. So when these two individuals mate and form another individual, that particular person will have one copy of this A from mother, another copy, say this A from the father. Okay, so that is how alleles get inherited.
Okay, ma'am. Thank you.
Okay. Now moving on to the next question. The question states here, which one of the following markers used in the human molecular genetics likely to show many allelic variants for a given locus in the normal population? First option is restriction fragment length polymorphism, or RFLP. Then another is variable number of tandem repeats. Then another is microsatellite. And the fourth option is single nucleotide polymorphism. So here, this question states that in human molecular genetics, okay, this is the subject that you're studying. So these options shows 4 types of markers that people generally use for identifying something. So these are 4 markers. So used in human molecular genetics, you have to identify uh, which of these markers will have a large number of variants, okay, within the same locus, and they should arrive, like they should be present within the normal population. So each of these are particular kind of markers for identifying a particular type of like say genetic condition or anything like that, for characterizing so like RFLP uses to identify where is a mutation, single point mutation has happened, something for characterizing some kind of genetic variation. Okay, now which of these markers will have large number of variants, and the variants will be located at the same position within the chromosome when studied in a large population of normal people. None of them have, disease. None of them are affected by a particular disease. Got my point? So using this understanding of all the 4 types of markers, you have to answer. Try, then I'll definitely say the correct answer, but you should think over it and try what will be the correct answer. So let me recall, okay, what do you mean by RFLP? What do you mean by SNPs? What do you mean microsatellites? So these 3 are discussed, right? Then there are tandem repeats. Let me recall, refresh your memories so that you can answer this question. Will that work? Okay. So if you remember, RFLP is a method, right? Molecular biology method, where because of point mutation within a genetic sequence, it has become a place where a restriction enzyme can cut, right? So there are 2 genes, one is normal gene, another is mutant gene. So you give a restriction enzyme like EcoR1C. Now, because of the mutation within the particular genetic sequence, EcoR1 can now cut at that particular position because a restriction site has emerged. So because of which, this is the normal one, normal gene, and this is the mutant gene. EcoR1 has cut here. So when you take these two genetic products and do, gel you run it in gel, you will see for the normal gene, there will be one large band, whereas for the mutant gene, which has been cut, a double strand break has happened because of EcoR1, you will find two bands. Like I'm arbitrary drawing. So that is how people identify the point mutations, okay, using RFLP method. Then regarding tandem repeats, they are actually used for fingerprinting, DNA fingerprinting, okay, which forensic people use, forensics, to identify who are the parents of a particular child, etc. Okay, and these are just like repeats like CA repeats or CAG repeats, which are found at a particular site within a chromosome that people know, and then they track in the child, in the mother and father. Okay, so not much about tandem repeats. Then there are microsatellites. So microsatellites are again repeats, right? Like CA repeats, etc. And CAG, these kind of repeats. And they are inherited from one generation to the next generation. They are present in affected people, normal people, in everyone. Okay, and this number of N varies. It can vary. So one person can have 5 CA repeats. One person can have 10 CA repeats. Okay, so people use these microsatellites for identifying the mutated gene in monogenic disorders. And then there are single nucleotide polymorphisms, which are nothing but base substitution that has happened at a particular site within the gene. And people have identified what are these SNPs, and people use it for tracking the polygenic disorders, and they are also present in both normal and affected population. But the thing is, SNPs at a particular location can be maximum 2, right? So this is the, say, DNA sequence, and you found A, okay, adenine, okay, in the mutated gene where a mutation has happened. Where the normal, it was T. So when this mutated gene is inherited, a complementary base pair will happen, then T only will come, right? So it can be either A or T, the SNP, right? Whereas by microsatellites, the number can be huge. Any number of repeats can be present. So now try to answer which of these markers will show a large number of allelic variants, means within the same locus, okay? So locus means the position, within the same position in the chromosome, you can say, or within the gene of interest that you're studying, and it is present in both affected and normal, in all population. You're studying a population, then it will be presented there. So now try to answer what will be the correct answer, like answer to this particular. Okay, you're correct. If anyone have any query on this, do let me know. The answer is microsatellite to this particular question, because microsatellites, it can have various number of repeats, right? And they are all present within the same locus in the chromosome. Fine, no queries. Now, as I promised previously, that I'll be discussing the positional cloning approach in more detail. And if time permits, I'll be discussing a word problem as well towards the end. Okay. So for doing positional, you know why positional cloning is important, why people have used this method, which I've already discussed. Now, how do people do positional cloning? What are the steps involved? First, if we are studying a particular disorder like hemophilia, etc., you need to see how this disorder has passed on from one generation to the next generation. So for that, you need to draw a pedigree and do pedigree analysis. Means you have to identify whether that disorder is autosomal or X-linked, Y-linked, whether that disorder is dominant or recessive. So you have to do that first. You have to first identify. After you are done with the identification, your next task will be to identify the position within. Say you identified that if from the first analysis you identified that the disorder is of autosomal type. Okay, means it will be present in all 22 pairs of chromosome. The sex chromosomes, it is not present, so you have ruled out that option. And that disorder is of, say, recessive type. Means the mutant allele will have to be present in homozygous condition, then only that disease will be present in that particular individual. So the second step, you will then look for the position within all autosomal chromosomes. Where you feel that the defected or the mutated gene could be present. Mutated gene could be present. So how you will do it? You will use the linkage, right? The concept of linkage. So you know what are linked genes, right? So the two genes which are present at two different loci and they inherit together. Similarly, linkage refers to analyzing the markers. Here, people use microsatellite markers, which are very tightly linked with the defected gene. So you don't know the defected gene, but you do know the macrosatellite markers. So if you can identify few macrosatellite markers which are inherited from one generation to next generation, and you see that in all the affected individual, the macrosatellite marker is present in the homozygous condition, then you can guess that within the portion of two macrosatellite markers, the defected gene is present. Okay, so till now, you can do this. Now, as I said, you have to do this for all chromosomes. So you will be studying hundreds of microsatellite markers, and you have to remove the option where you get results because of chance, okay, and not because of pure linkage. So for each microsatellite marker, you have to calculate what is called LOD ratios, which is a statistical formula is there. People just put the values. Okay, so the microsatellite markers which shows the highest LOD value, those microsatellite markers are chosen because people assume that only these markers will be closely linked with the defected gene. Rest of the markers may be passed on from one generation to the next generation by sheer chance, not because of linkage. Okay, so by LOD, you have narrowed down your options for microsatellite markers, and then you use these markers where you have found uses marker to identify the region of the chromosome. Okay, so by now you can also from 22 chromosome you can come up to one chromosome in where a small region is your target site, where you have where you assume where their candidate gene, the mutated gene, would be present. Okay, this is your second step. Now the third step. Now what you will do is walk, chromosomal walking, or physical mapping. This is the third step that you will do. Now, you will say you have in that particular region of the chromosome, you have found say 6 macrosatellites, and let's name them as A, C, D, E, F, okay, or S. Okay, so these are your 6 markers. And you have to identify what are the positions of this particular markers, means what is the order in which these 6 markers lie. And for doing that, you will be using genomic library, okay, and within the genomic library, which are not basically just the fragments of the genome, and within the fragments you will locate where is the uh, position of the each of these markers like A, C, D, E, F, and then you make a sequence. Okay, so this thing will become more clear when we'll do a word problem. And after you have identified the correct sequence of these physical of these macrosatellites, what will you do next? You will try to identify what all genes might be present within the positions within the positions of these macrosatellite markers. So for that, you will use expression sequence tags, or EST it is called, okay, and check their presence. So for physical mapping and as well as the presence of the gene, you will have to do simple PCR. The PCR result will tell you the final outcome. Okay, so do I make sense, or is there any doubt with any one of you? Is there any doubt in understanding what you mean by or how you do positional cloning? Is there any doubt? Then after you have say identified like one gene, you have to validate that. Okay, you have to validate using an animal model, maybe where you have created a knockout mouse where that particular gene is deleted, and you see what is the phenotype that particular mouse is displaying, and you then you compare it the phenotypes with the phenotype that is normally associated with the disorder that you're studying. So if you find one or few of the phenotypes similar, then you can say with confidence that yes, mutation in this particular gene or deletion of this particular gene is causing that particular monogenic disorder. Okay, so through this approach, you are just identifying the gene, then you have to validate your results as well. For that, you can use animal models, or even you can use in vitro studies, okay, where within the cell you are deleting that particular gene and seeing the morphological changes or functional changes within the particular cell. So these kind of studies can be done to validate. Is there any doubt?
Can you please explain again the chromosomal walking?
Okay, sure. So let me draw so that you will explain, you'll understand that better. So you have, this is say is the chromosome, and you have identified this particular region, and this in this region there are say 6 markers, 6 macrosatellite markers within this particular region. Now you have to know what is the order in which these 6 markers lie. What is the correct order? Because if you don't know the correct order, you won't be able to go back into the pedigree and track how it has been how it has been inherited. The order of these markers also gives you what is called haplotype, which is nothing but a set of markers which are known to inherit together because they are linked. Okay, they are linked, so they will inherit together from one generation to the next generation. And in the pedigree, you will be able to see that in all the individuals who are affected with the disease, they will have the particular marker in the same sequence. Okay, so for walking, chromosomal walking or physical mapping, you will have to use the genomic library, which is nothing but the fragments of the whole genome. Okay, and they are cloned into, say, a carrier like yeast artificial chromosome or bacterial artificial chromosome or bacteriophage. Okay, within these type of carriers, your DNA fragment of the DNA is present. Now you have to check using PCR in which of the uh, entire set of DNA fragments your marker is present. Okay, each of the 6 markers is present. Okay, so let's suppose you have identified in back in a 6 back. I'm naming it as B1, B2, B3, B4, B5, and B6. And using PCR, you saw that, say, for 6 markers A, B, C, D, E, F, G. Right, 7 okay. So you saw that in B1, A is present. Okay, using PCR, you will do the band electrophoresis, and from the band you will get to know. Similarly, B, you saw it is present in B5, C is present in B2. Then using this PCR data, you have to arrange. Okay, you have to arrange the physical order in which these 6 markers are found in the chromosome. So after doing that, you will see that, say, B is present in the first position, say D is present in the last position. Okay, or there is then C, then A, okay, F, E, F. So you found from the PCR that this is the actual order in which these 6 markers are present within the chromosomes. So this is, you do it by physical mapping or chromosomal walking. Okay, one important prerequisite for doing physical mapping is that all your DNA fragments, say this is B1, this is B2, this is B3. So all your DNA fragments which are present in the back, the bacterial artificial chromosome, they should have overlapping regions. Okay, they should have some overlapping regions, and this kind of library is called contig. Okay, so you should have a contig with you where the fragments is such that each fragment is have some overlapping region with another fragment, because then only you will see that, say, A is present in all the 3 fragments. Similarly, you will find B is present in only B1, not in B2, B3. So when you arrange these fragments, you will automatically get the correct physical order of all the 6 markers. Understood? Or is there any doubt?
Yes, ma'am. Yes, ma'am. Understood.
Okay, great. So I will, if time permits, I will solve a word problem based on positional cloning approach, which was given to us as a quiz actually when I was doing this course here at IIT Kanpur. Okay. So Professor Ganesh took the course, and I did that, and there was a quiz problem. And that problem is so good for understanding this positional cloning thing. Okay, if time permits, I'll definitely discuss with you all. I don't know how relevant that is, but I thought for clarity in concept, that problem will be very nice. So I'll do it towards the end. Okay. So this is the fifth problem, which states that the defective gene for finding the defective gene from monogenic disorder using the traditional positional cloning approach is a sequential process, right? So we have to follow one step, the next step, right? So there are steps involved in the positional cloning approach. So which one of the following, which one of the options identifies the very first step of this approach? I've already discussed. Can you guys answer?
Pedigree analysis.
Correct. You are correct. So the correct answer is pedigree analysis. First, we have to make a pedigree and analyze, right? That is the very first step. Another question. In the positional cloning approach, the co-segregation of macrosatellite marker with the disease phenotype in a family within an autosomal dominant disorder indicates. So the disorder, people have identified that it is an autosomal dominant disorder, and there is a macrosatellite marker which have co-segregated, means it is linked with the gene of interest which you are trying to find out, right? Unknown gene. It is linked with that unknown gene, and it has passed on, okay, with the people within the family, and whoever have got that gene and that macrosatellite marker is showing the disease phenotype. So which of these option indicates that this is the scenario? The macrosatellite marker contributes to disease phenotype. Is it true? Does the marker contribute to disease phenotype? No, right? Then the marker is located very close to the gene involved in the disease. Then is the marker repeat? I mean, macrosatellite is a marker, right? Is the marker repeat is expanding in every generation, or is it the risk factor for the disease? So which of these four options would explain this kind of segregation within the family for this particular marker?
Option B.
D or B?
B, ma'am, B.
B. Yes, you're correct. So correct option is B. It is co-segregating because it is closely linked or located very close to that gene of interest, which is actually mutated and causing the disease, right? That is why it is co-segregating. Then what do you mean by haplotype? Okay, this question asks you that. So is it a group of individuals testing positive for a specific antibody, or is it a molecular biology method used to track the inheritance of genes in families? Okay, or is it a set of markers that is physically linked and segregated together? And lastly, is it a marker representing the haplotype genome? So what do you mean by haplotype then? I've already discussed when we are talking about physical mapping.
Option C.
Yes, you're correct. So haplotype is nothing but a set of markers which are linked together and will segregate together, right? So one generation to the next generation, the same set of markers will be present, right? Now this is the eighth question. Now this question states the sequencing data of many individuals from a normal population revealed a single nucleotide change at two sites in a gene. There's a normal population and there are many individuals within the population, and all these individuals showed single nucleotide change at two sites. So the change in site 1, we are talking about the first site, it has the frequency of 0.2%. Okay? Frequency of occurrence of this particular change is 0.2%, whereas change at site 2 was present 2%. One was 0.2%, another was 2%. So based on our understanding, which one of the following statements represent a likely explanation for the given data? Option A, site 1 is a disease-causing mutation and site 2 represent a normal polymorphism. Option B, site 1 represent a recent change and site 2 represent an older event. Option C, site 1, a change in site 1 is a consequence of change in site 2. And D is change in site 1 is induced due to exponential change in site 2. So which of these options explain this phenomena? Okay. What do you guys think? What would be the correct answer? Read the question again, or let me repeat that for you. A sequencing data of many individuals from a normal population revealed a single nucleotide change at two sites within a gene. The change at site 1 was present at 0.2% frequency, while the change at site 2 was present at 2% frequency in a population. So based on your understanding, which one of the following statements given statements represent a likely explanation for the given data? So the data is that change at site 1 has a 0.2 frequency, 0.2% frequency, means within a population having 100 people, only 0.2 people, we can't say, but like hardly one people have that particular change. Whereas within 100 people population, only 2 people have that particular change at site 2. So frequency of occurrence for change at site 2 is more than the frequency of occurrence at site 1. So what do you think would be the explanation for this particular phenomena?
Option B.
Option B. Okay. What about others? What about others? What do you guys think would be the correct answer? One person told option B. Others, think over it. It's fine if you are wrong. Ricky, Arshi, why don't you guys give it a try?
Option A.
Option A. Okay. So there are two answers. So both of you are wrong. The correct option is B. Okay. So this change, right? We only know what is the frequency of occurrence of this particular change. We have not been given anything about how this change is affecting that particular person. Is it that that person has a normal phenotype, or is that person has a diseased phenotype? We don't know anything. So if we choose option A, it doesn't match with the what is given in the question. Okay. Then there is option C. How it is like coming that change in site 1 is a consequence of change in site 2, right? How these two sites are changes in these two sites are related? Are we given in relation in this particular question? No, right? So option C is also not correct. What about option D? Are we told that means the exponentially the change has come in site 2, 0.2% and 2%. There is no exponential relation between the frequencies. But what we can see that frequency of occurrence of change in site A is much lower than frequency of change at site 2, right? So we can only say that this site at site 1, the change at site 1, it has occurred for the first time, like which is a new mutation which is yet to be propagated within the population. Whereas site 2 change is much older than that. It has come maybe 5-10 generation previously, and it has seeded within the population through mating and all. So to the percentages, we can just only say that it has come early, one change has come early, and one change is quite old. Okay, so the correct option is B. Did I make sense? Or anyone has any doubt in understanding this particular question?
Yeah, if it can be repeated, it will rather clear up the confusion which I have.
Mm, can you clearly tell your query again? I couldn't understand.
Can I again repeat? Then it would be easier to be understood.
Okay, so what I'm trying to say is that we have to, what is given in the question, we are given only what is the frequency of occurrence of the change in site 1 and the frequency of occurrence in of change in site 2 in a given population. We are just given the frequency. What do you mean by frequency? In how many people you see that particular change, right? That is a frequency. So we're given only that. And from this given data, we have to come up with a possible explanation, which could basically help us understand like why this particular change in frequency has happened, right? One is one change has 0.2% frequency, another change has 2% frequency. Okay, one is more frequent, one is less frequent within the population. So we have to come up with the suitable explanation. The correct answer is since the frequency of occurrence for the change in site 1 is lower, we can only guess that it has come up recently, okay? Like 1 or 2 generation, it has come up. It has not yet got the time to propagate within the population like through mating and all, okay? Whereas the change in site 2, you see that more and more people are having it, right? It is greater, relatively greater, 2%, right? So we can say from that, so this mutation, so these two mutations are say de novo, right? De novo in nature. They have come up within the population through a new mutation, novel mutation. And a second mutation, it has come previously, like 4 or 5 generation back, it has come up. And now it has propagated itself within the population, because of which more frequency is there. So that's why so the B option is a more clear explanation of why this thing is happening within the population. Not A, not C, not D. Neither of them explain it. Since we are not given anything about the phenotype of the individuals, do we know are they diseased or are they normal, right? Also, we are not given any relationship between these two types of changes, right? We don't know whether one is a consequence or one is the cause of the other. We don't know how change in one is causing the change in the another. We don't know these things. That's why all the other three options are not correct. B is the correct answer. Is that clear now?
Yeah, but like we know that the mutation is a random change, permanent change in the nucleotides, and it can only be a random or it is de novo in nature. So it affects the individual and its protein function, and so the machinery, and ultimately the phenotypes come up. So it is a mutation, right? So in case of normal population means the mutation, if it is present in the normal population, it will not harm the population in general, but it can lead to variation. So which is the path of evolution, right? So it is not harmful that much. So that's why it is variation. So the first option is also a confusion between the A and B, because the first is a disease causing mutation, because mutation tends to become the frequency is very much less compared to the normal population, which tends to be more frequent than the diseased one. So there is a confusion, although whatever explanation you have given correct, means it's easily could be understood, but there's a confusion for people like us who have not been involved in this kind of research activities frequently, but they can think that this can be an option.
Okay, so you are referring to the difference between what is a mutation and what is a polymorphism, right? Mutation only occurs in the affected individuals, and polymorphism seen in normal people as well. So, and we also know that mutations which are disease causing, they're the frequency is very less. You're referring to that explanation, right? And whereas if it is present in more than 1% of the population, and then it is a normal polymorphism event. So that is why you're referring. So to get clear, this in this question, first of all, we are not told what is the phenotype of the people having change at site 1. Okay, since we don't know the phenotype, we can't easily come to the conclusion that it is a disease causing mutation. Okay, so if we can't come to that conclusion, the second thing, that is it site to represent a null polymorphism, that conclusion is related to this one. So it is not possible to go to that conclusion. So when you know that this particular mutation, so here in this question, whoever is doing, they don't know the effect of this mutation. So if they later on find out that yes, this mutation is causing that particular disease, or they found that normal people having that particular mutation causing a disease, that then we can say that option A is correct. But since we don't know anything about what is the phenotype of that individual, we're just told there are individuals within a normal population, we are just told that. So disease thing is not coming at all. So maybe this particular point will help you distinguish between option A and option B. Do I make sense?
Yeah. Otherwise, the concept which I explained is correct, right, or wrong?
Yes, yes, yes. That option is correct and is applicable in many cases. So yeah, you're going in the right direction, but you have applied here where didn't need to be applied. Okay, so it can become a bit confusing. It's fine. Okay, you're just learning. So this is the 8th question. Now come to the 9th question. So this question is about gain of function mutation, which we discussed in the last week, right? What are gain of function mutation? And you have to identify which of these are the causes or could be the cause of gain of function mutation. So option A is yeah.
Should be
You okay? You're saying the answer is all of these. Sorry to interrupt. Can you repeat your answer?
yeah, all of this.
Yes, you are correct. So I would like to know, how did you come to this conclusion? Why it will be all of these? So that others would understand. Yeah.
Because balance translocation, we know gain loss of loss of mutation. It is it can result it ultimately in a truncation of peptide in whatever way it could be frame shift due to addition deletion, or it could be due to nonsense mutation, right? Whereas in case of gain of function, that's not the case. So it can be dinucleotide repeat expansion. Gain of function leads to novel phenotype, novel gene function, or it can be a loss of function like androgenic receptor. So it can both go other way, both go both can lead to uh, gain of function as well as loss of function. So gain of function uh, can result from any one of the criteria mentioned above. So that's why balanced translocation can also be gain of function as well. Balanced translocation, like in case of Prader-Willi Angelman. Sorry, in case of miscarriages. So both can lead to novel function, which can be harmful as well as it can help to add some benefit to the person or the population. That's why it would be all of this.
Okay. So adding to what Ricky said, I would say that any of these types of mutations that are given, right, the causes of mutation, uh, you don't know to which kind of gene it is affecting. So wherever that mutation has happened, whether it is translocation or recombination or incorporation of particular error, wherever it has happened, the gene, okay, now subsequent result of this mutation will decide whether the mutation is a gain of function or loss of function mutation. Okay, so the type of mutation ultimately depends where the mutation is happening, depends on the location of mutation, okay, within the gene. So since we are not given any information, right, about what is where the mutation is happening or anything, but all of these mutational events, right, exchange of DNA segments happen, and depending on where the DNA segment is added or getting deleted within the gene, we can have a loss of function or gain of function. So if the question was stated otherwise, like a loss of function mutation can be caused by, then also the answer can be all of these, right? Uh, because depending on how the resultant gene or mutated gene is going to behave, you will get to know, right, whether it is gain of function or loss of function. Do I make sense? This explanation is this explanation clear to you all?
Yes, ma'am.
Okay. Now moving on. So there are three things, okay, which have come up after the human genome has been sequenced completely. One is genomics, the area of genomics, where people study the genetic material, right? The DNA, which contains all the information about how a living organism should function, right? Then from this DNA, within the DNA, there are, or within all the chromosomes, there are some coding regions, there are non-coding regions. The coding regions, they give rise to mRNAs. So set of all mRNAs within the cell, okay, and those are studied in the field of transcriptomics. And from these mRNAs, we know through the process of translation, protein is made, right? So within the cell, the set of all proteins are studied in the field of proteomics. Now, all these three sections, okay, they have their own tools how to do that, okay. People have developed various methods. So genomics is very well established, okay, and proteomics and transcriptomics, these are coming up fields, okay. And one thing about genomics is that it is people study about data which is static in nature, means until and unless there is some mutation due to uh, mutagen or anything, the DNA content is static. Okay, it is DNA, we know, is very much stable in nature. But yes, well, say in 1 million bases, there is a chance that one base is mutated or changed, right? So the DNA is a very, very stable structure. So it is static in nature. So the people study the static information that is present in the DNA. Whereas from within the DNA, complete DNA content of a cell, there are few regions that is called genes, which are transcribed into mRNAs. So at a particular point of time, okay, particular point of time, a few mRNAs will be produced. Then after few years, expression of few other genes will be favored. So this the transcriptome data within the cell, it changes with the age of the person. Okay, it can also change with the environment. So one environment favors the expression of particular genes for the survival of that individual, say in cold environment. Whereas people who are living in tropics, they will express some kind of genes that favor the survival in the tropics. So even the environment affects the what will be the total mRNA content present within the cell at a particular time in the individual's life. Okay. Now, how this mRNA is translated and made into proteins. This collection of all the proteins is studied in the field of proteomics. Now, it is possible that from a particular gene, say gene, 10,000 mRNAs are produced. Okay, and now within this 10,000 mRNAs, few have didn't transcribe properly. Okay, so there are proofreading mechanisms which can read the mRNA sequences, and found that only 1,000 of these should get translated. Okay, now this 1,000 mRNAs will go into translation process. Now, while translation, there is some defect. Okay, within the so some wrong amino acid gets added within the polypeptide. So there's mechanism which degrades such kind of defective polypeptides, and the amino acids are again recycled for making of any other polypeptide chain. So from this 1,000 mRNAs, say we get only 100 proteins. So this is how the cell actually also regulating each step, right? Now within the 100 proteins, few proteins, say half of them, they are functional in their native form. Okay, whatever the result of the translation process, the protein has been made, they'll be used as native form. The rest 50%, they can undergo post-translation modification. So what I'm trying to say here is the mRNA content, total mRNA content changes, right? Within the cell, it is dynamic. Similarly, the total protein content also changes within the cell. And it again changes with the age of the individual, it changes with the environment. Okay. And also these proteins, are the effectors of all the things that is happening in the body. Hormones, cell membrane receptors, anything you say, enzymes, etc. These are all proteins in nature. So so the production of protein is happening at every organelle within the cell, and like hundreds and hundreds of copies of such proteins are being made. The collection of all the proteins, these are studied in the field of proteomics. Okay, and how this post translation is affecting the activity of particular proteins, these are also studied there. Okay, so with this short primer, I would like to ask you guys to look at this question. So you are provided with the genomic, transcriptomic, and proteomic data of two different tissues, tissue A and tissue B, from a patient. So from an individual, you have got two tissue types, tissue A and tissue B. Say one is skin tissue, another is liver tissue. Okay, so they are from two different organs or two different types entirely. So which of the following data will be most similar for these two tissues? Whether genomic data will be similar, or transcriptomic data will be similar, proteomic data, or in all of these tissues, all three types of data will be similar. So what do you think will be the correct answer?
Genomic data.
Correct. So as I told, genomic data is static in nature, and all the cells in the body, they have the same genomic content. Okay? That the embryo has formed, and the chromosomes they get from the mother and father. The same, mitosis process happens. They get multiplicated and then divided in daughter cells. The genomic data is exactly the same. This is the next question. Which one of the following statements is true? All DNA variations are mutations. All mutations affect survival. All variations are in transgenic regions. Some variation may increase fitness. So which of this option is true? Think over it. Like which of these options seems the most correct according to you? Let me repeat. So there are 4 options given here, and you have to identify which will be the most correct statement to you. What do you guys think?
Gain of function sometimes.
So gain of function is not given in the option. Can you say whether it is A, B, C, or D? Which one is the correct answer?
I think D.
D. Okay. So you are correct. Some variations may increase the fitness. Okay. And also not all variations are actually mutations. By mutations, I mean those variations which lead to particular disease. Okay. So those particular variations are people called mutations. Okay. Then all mutations affect survival. Again, this may not be correct. So there are sometimes some mutations which are present in all normal population as well. And it doesn't have to happen only in the intronic region. It can also happen in exonic region, the variations. So D is the correct answer. Now moving on, what do you mean by genetic risk factors? Okay, so these 3 things: genetic risk factors, SNPs, association studies that people utter. These terms people utter when they are trying to study or trying to explain any polygenic disorder. Okay, so polygenic disorders are those disorders which are due to mutations in more than one gene. Okay, and people have found through HapMap project there are few common variations which are present in our population. So I think there are 10 million common variations that people found in the HapMap project. So these variations they termed as SNPs, a single nucleotide polymorphism, means a change in a single base is there, or point mutations, right? So 10 million SNPs people have found, which are commonly available in the world population. Okay, and what people have seen that there's a group of individuals, okay, and they have a particular disorder, say diabetes. So diabetes is a disorder which comes under the polygenic disorder kind of a category. Okay, these individuals have the diabetes, and another group they don't have the diabetes, no diabetes. So the people who have the diabetes, these group will be called as a case group, whereas the group where there is no diabetes, marking it with red, they will be control. So only difference between these two population is whether the group has diabetes or not. Rest, the age, then ethnicity, okay, etc. These are all similar. Even the male to female ratio is also similar within this group. Male to female ratio, it is also similar. So people try to see what SNPs, okay, are present in the case group, okay, and whether these SNPs are present in the control group or not. So this type of study is called association study, and this is a statistical method. It uses a statistical method in order to associate or find a relation between a particular allele and a particular phenotype, disease phenotype. If they find that and these type of association studies involves a large population, okay, they study a large group of people for while doing this study, then only statistically they will get some results, good results. So they try to see if particular SNP, if it is present in say 100 people with diabetes, and if it is not present, okay, in a group who doesn't have diabetes, then they can say with some statistical backing that this SNP is likely to cause this particular disease, that is diabetes. So if this person, say any other person A, if this person A have the SNP that has been associated with diabetes, this person A will have higher risk of developing the disease. Okay, so SNPs which have been identified, or variations which have been identified as being associated with a particular phenotype. Okay, and if these SNPs are present in some population, we can if present in a particular individual, we can say that they are risk. Okay, they are a risk factor that in after few years maybe, or their children of that individual will have the disease, that is diabetes. So complex disorders, within complex disorder, we always say there's a likelihood. Okay, it will possibly cause it. We cannot say that it will definitely cause with confidence, because we know if there are multiple alleles are present. Okay, and to what degree each allele is contributing to that particular disease is still being studied. So only we can say the presence or absence, whether it is going to be a risk or no risk thing. So that is all about association studies and what are SNPs and what are immunogenetic risk factors. It is often said that if an SNP within a population is found on less than 1% of the people, so it is not a risk factor. But if it is present in more than 1% of the population, and they have seen that in their previous studies that this SNP is linked or associated with a particular disease, so they can say that it is likely that the population having that particular SNP will be having, or they are prone to having the diabetic disease. Now, there's another concept that I would like to explain, is called linkage disequilibrium. Linkage disequilibrium. Now, if any of you are reading that book by Tom Reed and Tom and Reed, right? That Human Molecular Genetics book. So there they have beautifully explained what do you mean by linkage disequilibrium. Okay, so what happens when people are studying? When people are doing this association studies, they don't take into the factor that two individuals within the population, they can be related in any way. So all of the individuals who are in the case of the control group, they belong from different families. But sometimes if they trace whether or not they have a common ancestor, say 25-30 generations back, if they have a common ancestor and that particular ancestor had a mutant allele, okay, and this mutant allele has somehow passed on after 25 generation to his descendant, and now these descendants are not related to each other if we look back to 3 or 4 generations. Okay, so when in these kind of scenarios, linkage disequilibrium comes into play, because it uses the concept of linkage. Means when the marker is closely associated with the mutant allele, and they are transmitted from one generation to the next generation together. Okay, and when this kind of thing happens in a large population where the individuals are not related, when you study their generation up to 4 and 5 generations only, okay, in those people. But if you see very distant, like 20-25 generation back, that their ancestor 20-25 generations back, their ancestor had that linked gene, okay, to which a marker is there, and you are able to trace that marker within the individuals today in the current day, then you can say that the association of a particular variation to that particular to a particular disease is due to linkage disequilibrium. Okay, so this linkage disequilibrium can be cause of association between, say, allele A and disease T, given the case that the people in question, they had a common ancestor, but this ancestor was like very long back, okay, say 5,000 years ago. Okay? So if this kind of thing people have found out, then they can say, or they can conclude that the association between allele A and disease D is because of linkage disequilibrium. Okay, so till now the concepts, are we clear on that, or are we having any doubt? Is it clear? Okay, I'm assuming that it's clear. So this is a question for you guys. In polygenic disorders, the term genetic risk factor refers to. Okay, so what do you mean by genetic risk factor? First, it is a disease associated gene variant inherited from parents. It is a gene variant that confers a small yet significant risk of developing the disorder. It is a gene variant that is defected in the normal population. A D. Is it all of these? What do you guys think is the correct answer? Arshi, Anju, do you guys would you like guys like to try?
Option D.
Option D. So you are wrong. The correct answer is option B. Okay, because a genetic risk factor doesn't it can be one of the alleles risk factor that people have identified. It can be one of the alleles among various other alleles that actually together cause a polygenic disorder. So its presence confers a risk. Okay, it doesn't with confidence say that the population or the person who is having this particular mutation will have the disease. Okay, so it gives a probability of risk, okay, that the person will have a particular disease, polygenic disorder. Okay, uh, are you clear on this, why it is option B?
Yes, ma'am.
The next question, which one of the following statement distinguish the genetic risk factor involved in a polygenic disorder from the mutant allele causing a monogenic disorder? So you know there's a mutant allele, right? Is a mutant gene which is causing a monogenic disorder. There's only one gene. Whereas polygenic disorder, there are various kind of alleles, and each presence of each allele can be said that that person is having the risk of risk of developing that disorder. So you have to distinguish between these two things: hereditary risk factor and the mutant allele. So mutant allele is a DNA variant, while a genetic risk factor is not. The frequency of risk factor is likely to be higher in the normal population as compared to mutant allele. The risk factor is a normal gene, whereas the mutant allele is a defective gene. Risk factor refers to SNPs, while the mutant alleles refers to deletion. So what do you guys think will be the correct answer? Which of these statements distinguishes between these two things? Risk factor and mutant allele. Ricky, would you like to try?
I think Genetic risk factor involved in polygenic disorder.
Yes, can you repeat?
Genetic risk factor in polygenic disorder would be small, and mutant allele causing monogenic disorder. Bit tricky, I think, this one.
So you actually saying exactly the opposite of what we were discussing in one of the previous question about what is mutation and what is polymorphism. The genetic risk factor actually refers to the polymorphism thing, which is normally present in the population.
Yeah. So I think C.
Okay, so you're wrong. The correct answer is B. Why B? Because the risk factor, as I said, is a polymorphism, and in the normal people, the frequency will be obviously high, right? Uh, because the presence of risk factor doesn't guarantee that the person will be having a disease. It can be present in our population who is healthy, not displaying any disease phenotype. As a mutation, if since we are talking about monogenic disorder, whoever is having this mutant allele will be a diseased person, and the disease may be such that it doesn't allow the inheritance to the next generation. The person dies very early age, so because of which the frequency of this mutant allele is decreased. Why not C, which you said just now? So risk factor is a type of mutation. Think about it. It's a type of mutation, right? Uh, but it is not affecting the function of the gene. Okay, so gene, the normal gene, the normal gene, the term normal gene is very unclear, right? And defective gene. Okay, fine, mutant allele is having some deletion or some substitution, anything because of which it is not producing a defective product. Okay, means it is producing defective product, and because of which disease is happening. But risk factor still introduces, risk factor is still some kind of a mutation, and sometimes the risk factor doesn't have to be within the coding region. It can be elsewhere as well, if I'm not correct. Means it can be non-coding region as well. Let me check. Am I correct? Take risk factor in non-coding region. Yeah, so it can be in the non-coding region as well. Okay, and also any mutation in non-coding region can also lead to a disease. So what I'm trying to say is risk factor it is not affecting the function of the gene. Okay, so because of which the function is just like normal. But if you compare it with option B, the frequency, in terms of frequency, option B seems more accurate to me. Whereas the other A and B options, these are obviously incorrect, right? So mutant allele is a DNA variant, where genetic risk factor is not. So both are some kind of versions of a particular gene sequence, right? And this only refers to SNPs, and this only refers to deletions. Yes, yeah, it refers to SNPs, but mutant allele doesn't have to be only because of a deletion event. It can be happen due to other mutational events as well, right? Which you're all familiar with. So between B and C, there is a bit confusion. Let's see, but the correct option is B for this because of the frequency thing. What do you guys think? Do you guys also have a bit of confusion between B and C? Or is it just me and Ricky over here? Samyu Gupta, right? Did I spell your name correctly? Shamyukta. Acha, Shamyukta. Sorry. Shamyukta, what do you think will be the correct answer? Do you also feel there is a confusion between option B and C? Or do you have any other explanation for this particular question? Uh, yeah,
Yes, ma'am.
yeah. Can you repeat?
I I think option B is right, ma'am. I don't think C.
Okay, so option D, I said that mutant allele, formation of a mutant allele doesn't necessarily has to happen through deletion. It can happen through other mutational event as well. A single base pair substitution or insertion can also lead to a mutant allele, right? So because of this particular point, the option D is void, doesn't make sense, or is not applicable to this particular question. But what I'm confused, like Ricky, is between option B and C.
Yes, ma'am. I told option B, ma'am. B.
D. So why do you think D is the correct answer?
No, ma'am. Option B. The frequency of genetic risk factor is higher in.
Okay. Sorry, I was hearing D. It's fine. So according to her as well, B is the correct answer. So one point just came to my mind regarding the how the risk factors work is okay, fine. So if there are 5 risk factors or the 5 different kinds of variants involved in a particular polygenic disorder, okay, and the person who is having all the 5 variants uh, will definitely show that disorder. But uh, if another person is there who is having only 3 of the variants, right? Together, when these variants work, they will show the disorder in some another form. So a person having all the 5 risk factors will show a more severe symptom, where the person having 3 having less severe symptom. So when risk factors together are coming, okay, in an individual, they are showing an additive effect, okay. So in that case, the risk factor are no longer acting as a normal thing. It is no longer a normal gene. Got my point? Um, and this thing that 5-3 this have been established through studies, and this is for any disorder, okay, like XYZ disorder, hypothetical disorder. But what I'm trying to say that the risk factors, they have an additive effect, and because of which the symptoms can be different. That's why it becomes very difficult for people to study these complex disorders or polygenic disorders. So when we are looking into that perspective, genetic risk factor are no longer normal in those cases. Ricky, what do you think of this another perspective of looking into this problem?
If I can think about the risk factor definition of it, it is a small portion, yet it is significant part of the population. But anyway, the risk factor is normal, as you said, that it is related to the polymorphism, which is kind of variation presenting present in a normal population, whereas kind of mutant allele, it has already established that it is a permanent change in basis. It should be small. I think, yeah, I mean, these two options, it could be either way correct, but if we have to select one, then there's a problem.
Okay, think about it. Okay, let's move on to the next question now. Which one of the following options truly represent the advantage of single nucleotide polymorphism as markers for identifying genetic risk factors, okay, for polygenic traits. First is they are amenable to high throughput screens. They are commonly present in the normal population. They are spread throughout the length of the chromosomes, hence give better coverage. All of these. So basically, you have to tell which of these options are advantages of using SNPs as markers for identifying the risk factors in polygenic disorders. This should be simple enough. Would you guys like to try?
All of these.
Yes, you're correct. All of these. This was a simple question. So next question is here. In a typical case control study, means where this study is used for studying the association between the gene and the phenotype, the subjects in the case group are expected to differ from the control group in what sense? Age, ethnicity, genetic makeup, or lifestyle?
Genetic makeup.
Correct. So last two questions were very simple. So as I told, there was an international HapMap project which was started right after people have sequenced the whole human genome. So in this project, people try to identify the common variations which are found in the populations. So they chose population from three regions: Asia, Europe, and Africa. Few populations from these regions, these three geographic regions, and identified what are the common variations present within their genome. Okay, and tried to do association studies to check which of these variations or markers for this variation, that is SNPs, are more common uh, um, in people who are having a particular type of disorder, polygenic disorder. And people also identified haplotypes, which are nothing but the genetic sequence which is identified as the risk factor. Okay. So haplotypes are again the markers. Okay, here the markers people using are SNPs, which are linked together, and they are segregating together when they are passed on from one generation to next generation. So here also for identifying the genetic risk factors, people used inheritance of haplotypes and checked how that particular genetic risk factor has inherited within families and in between in unrelated families. Okay, so this is one question about HapMap project. So what is the mandate of the international HapMap project? First, is it to define the haplogroup of the mitochondrial genome? Was it to map the whole human evolution, or was it to develop the tools for comparative genomics, or was it to find out the common patterns of DNA sequence variation in the human genome? So what was the mandate for the HapMap project?
It's D.
Yes, you are correct. It is option D. Okay. So in HapMap project, they found what are the common patterns of variations, means common variations within the population of people. You know? Now, which one of the following statements lists the goals? This is again a similar question. Okay. Similar list the goals of HapMap project. First, to determine the common patterns of DNA sequence variations in the human genome, to identify common variants likely to be important in the risk of common diseases like lifestyle diseases, etc. Then to make genome related information freely available in the public domain, and all of these. So it is just like this previous question.
D as well.
Yes, Ricky, I couldn't hear you. Can you repeat?
Yeah, it's D as well.
Yes, the correct option is D. All of these options were the goals for the HapMap project. Then in the HapMap project, the term linkage disequilibrium refers to what? I've already explained what do you mean by linkage disequilibrium. It means the variants, right? Or the SNPs, the markers associated with the mutant allele. Not, I shouldn't use that term, genetic risk factor. Okay. So whatever SNP linked to the genetic risk factor, they are segregated from one generation to the next generation, and it is not happening randomly. It is because of the linkage between the SNP and the genetic risk factor within the population. And it happened because there was a common ancestor within the two individuals in question. Okay, so this linkage disequilibrium has been found out as a cause when people are trying to associate the particular allele or genetic risk factor with the particular disorder. So in this question, we have to identify what is the definition of linkage disequilibrium then. Is it a non-random association of alleles at different loci, or is it random association? Or is it linkage of alleles with a given allele, or it is non-linkage of alleles with a given allele within a given loci? What do you guys think is the correct definition of linkage disequilibrium? So here linkage of alleles within a given loci, it refers to the within a locus, as I drew in this slide. I think this is one loci, right? This is one. Within the one, there are two alleles. So option C and D, they were referring that these two loci are linked. But can it be linked? No, right? They cannot be linked because during recombination, crossing over happens, and then subsequently the two homologous chromosomes get separated while gamete formation. So within the same loci, or within the same locus, sorry, the alleles cannot be linked. So linkage always refers to, which one this is? Linkage always refers to, or linkage disequilibrium always refers to linkage between two alleles which are at different locus. All right. Now here the randomness and non-randomness, that is the only question. So what do you guys think is the correct answer? A. Okay. So you are correct. It is A. This linkage, it doesn't happen if there is a possibility of randomness, people remove that possibility and only look into non-random thing. Means it is bound to repeat over and over again through the generations, okay, or within the populations. So linkage disequilibrium refers to non-random association of alleles at different loci. Okay.
So, but here the definition definition of the linkage to this disequilibrium is the degree of which one allele of one gene variant is related or co-inherited with the other variant of the allele within the same population, but the option says different loci. Why?
So I think you have found this definition through Google, right?
Yeah, it was there in the assignment only.
Okay. So I just opened it. Let me read it once. So here linkage disequilibrium describes the degree to which an allele of one genetic variant is inherited or correlated with an allele of a nearby genetic variant within a given population. Right? So here actually refers to that linkage thing only. So there is one allele Sorry, let me draw it like this. This is homologous chromosome. This is one locus A1. This is another locus 2. So here this is A. Okay, A is a gene. Allele is there of A. Is present on another homologous chromosome B. Then at locus 2, there is a gene B. Its allele is there, which is represented with small b. Okay, so it is saying that how much degree means, what do you mean by degree? Degree to which means in how many times people have observed that A, say A, this is one genetic variant, is inherited or correlated, means they are connected. A is connected with B. Correlated means there is a correlation between them, or how frequently these two A and B, they are inherited within a given population. Means for them, for A and B to be inherited together, they has to be linked. Okay, and these linkage, if people are saying more and more within the population, then it cannot be a random event. Okay, it has to be a non-random thing. So whatever statement that is given in the assignment, if we reframe that particular statement, then this statement, which is given in option A for question 18, it translates to this. Okay,
So why is different loci it is mentioned here? It should be in the population setup.
so see, for two alleles to be inherited together, they have to be a different locus. Try to understand. So this is locus 1 and this is locus 2, right? So within the same locus, this is homologous chromosome that is coming from mother, other is coming from the father, right? So these two chromosomes, in normal condition, they cannot inherit together into a single gamete. Okay, in abnormal case, that is non-disjunction, where that can happen, but in normal case always these two homologous chromosomes separate and get distributed within the gametes, right? So in normal case, these two alleles, they cannot be inherited. They always have to, if for inheritation together for any two genes, these two genes have to be present at different locus. Now the position of these genes should be such that there should not be any recombination happening between A and B, which separates them. Okay, so that is the linkage thing. Linked genes, these two has to be linked genes. So that's why the term different loci is used, and these two are non-randomly associated, right? Means people remove the possibility that these two genes A and B might be happening or might be coming together to do a random process or random event. So for positional cloning, they calculated LOD's ratio. So here for HapMap project or for association study, they also determine few statistical values, okay, to remove that random behavior. They only look into the non-random thing. Okay, means that is bound to happen over and over again. Do I make sense, or is there any other doubt?
Yeah, it was clear, but why there should be, I think, while reading up the definition of the linkage disequilibrium, so it says this defined loci of the same gene, right? The different gene.
No. Allele A and allele B. These
Yeah.
two represent the same gene. They are the variants of the same gene.
Okay.
Variants of the same gene, they are always present at the same locus. Okay, so now it must be clear, right?
Yeah.
So we are almost done with the time, but I wanted to discuss this word problem with you guys. Okay, you guys can take a screenshot so that you guys can read this problem later on, but I will quickly explain how to solve this particular word problem. So here in this problem, you are given a pedigree like this, okay, of a family, and this is the this person is the index case who has come to you, okay? And in this pedigree, you have to identify the unknown gene using positional cloning approach, which is causing the disease, okay? One in individual 1, 1, 1 and 4, 1. So while looking at the pedigree, you will understand that this is an autosomal recessive type of inheritance is going on, because there is consanguinity. Also, the affected children don't have affected parents, right? It affects both male and female. So from this pedigree, we can say that this is autosomal recessive. Now, you have to do genome work. Please take a screenshot of this image of this problem. I'm just quickly going through how to solve this problem so that later on you can follow. This video will be also available to you guys, so that later on you can refer to whatever I'm explaining. Okay, so you have to do a genome work using PCR approach, and you have been given these 6 markers. Okay, and sorry, you have been given this 6 genomic fragments in form of bacterial artificial chromosomes, and within these genomic fragments you found out that the markers S, D, W, N, G, A, how they are present. Now using this table you have to identify the physical order, or what is the correct order, or what is the sequence of these markers within the gene of interest. So how you will solve this problem? Let me take a blank slide. Okay, so here you have been given these 6 markers. Let's consider the first fragment. It is not working. So let's consider the first marker, B1. Okay, in B1 you see that these first fragment B1, you see that S, D and A are present. So let's assume S is here, D is here, A is here. Okay, this is D. Assume. Okay, then in B2, D and A is here. So if you draw a fragment like this, D and A is here. Then in B3, there is S and M. Here is S, you have written. M will be somewhere upstream of S. And B3 ho gaya. Then there is B4, there is D and A, right? D and A. So like this, you have to mark what could be the position of these markers. B4. Then there is B5, S, M, and A is here. B5, S, M, and A is here, right? S, M, A. Then there is in B6, there is D, W, B6, D, W, then G. So W and G, has it come up previously? No. G, I think I missed marking G. B2 mein D, G, A was there. D, G, G could be upstream. D, G, A. Then there is B6DWG. D, G, and somewhere upstream it can be W. So when you look at this, right, you can get the final sequence. It is M, S, D, then A, G, W. Okay. Are you guys following? How have I how have I physically ordered?
Yes,
Is
ma'am.
it okay? You guys can do it yourself. Okay.
But the ultimate fragment generated, what would be the order of it actually? Is it the same one, whatever you have?
Yes,
Just
yes,
MS
yes,
AGW?
yes. MSA, MSA, MSDAGW. Okay. So this is the ultimate sequence. Let me clear the above and write it nicely. So M, S, D, A, G, W. Or we can also write it as W, G, A, D, S, M. This could be another way of writing the order of these markers. Okay.
But just the reverse order.
Yes, yes, yes. It's just a reverse, same thing. We can read in the front or in the back, reverse order. It's the same thing. Next, we were asked in that question to identify the haplotype. Okay. That could possibly, or the minimum region that could possibly harbor the disease gene. Now, since we have gotten what will be the order, here this is the haplotype given. See, we have to write these markers in the order which we have obtained. So I'm doing, let's say, for individual 1, my obtained order is W, G, A, D, S, M, or you can do ulta as well. It doesn't matter. So for W, it was 56. Then G, it was 81. 81. A was 89. D was 35. S 27. M 42. Okay. Similarly, you have to write for all the individuals. Okay, and complete this pedigree. So this is a task when you do it on your own. You have to write the sequence. You will find a region within this sequence where homozygosity you will find. So let me quickly write the answer. So you will see in individual, this is individual 1, 1. You will see in individual 4, 1. This guy, for this guy, we will see that at position W, G, A, D, S, M. At position A and D, there is a homozygous condition of 55 and 66. You'll able to get when you have rearranged these markers in the same physical order which you have obtained from this question. Okay, so in this question, you will find that in this minimum order, in this minimum region, you will find the defective gene or gene of interest, which you were finding, right? Which you were trying to find, and then you have to identify this gene. C part, you have to identify this gene. So for identifying the gene, you will be using the second table, this one, where the PCR results for expression sequence tags is given. What is expression sequence tags? These are basically the probes for each of the genes, G1, G2, and G3. And they are trying to check whether these probes are present in these bags, B1, B2, B3. So how we will do that? Let me remove. Again, we will write each bag at a time. In B1, they saw that G1 is present. Okay. And from the previous discussion, from the previous table, actually, this table, which was the PCR results for the microsatellite markers, from that table, we knew that in B1 there was S, D, A, and here G1 is present. Then there is B2, right? From the previous discussion, we know that B2 has D, A, G, and the second table, B2 has G1. Okay. B3, which is another fragment, we know from the first table that it has S, and then this side upstream M is there. But in B3, there is only G2. Then there is B4, another fourth fragment. It has D and A. This fragment is just like this. And this fourth fragment, D and A, it has G1. And last B5 and then B6. B5, we had S, M, A. S, M, A. I think I have done an error in physical ordering, which I'm just identifying just now. The correct order would be, again my stylus is going. So correct order will be, here it will be, in the stylus. Here it will be A and here it will be D actually, okay, which I have missed in my previous order. A, D.
Ma'am, I have a doubt.
Yeah, sure. Tell me.
How do we find that M is upstream of S and B3?
Yeah, can you repeat?
How do we tell that M is upstream of S?
Okay, so see, if M is lying here, okay, in between S and A, right? Then this B1 should also have M here, right? So in B3, compare B3 and B1. If M, say, was downstream of S, then B1 fragment should also show positive for M, but it is not present in B1. So we have to assume otherwise that it would be upstream of S. Got my point?
Okay. Okay, ma'am, got it.
Okay. So that was the mistake I did. For B6, B6 नहीं लिख रही थी, I was writing B5, S, M, and A. So for B5, S should be here, M has to be here, and A. Previously, what mistake I did was S, M, and A. Right. So previously what I did was I wrote the order in such a way for the fragment B5 that it was, when we compared with other fragments, the marker was coming in between. Okay, so that is why the order is coming to be wrong. So now I have corrected that order. And lastly, B6 is D W G, here D W G, so G and upstream. W can't be towards the, sorry, W has to be downstream, it cannot be upstream, because if it were upstream it would have appeared in previous or above fragments. So what we saw that in B5 G2 is here, right, and in B6 G3 is there. Now if I take another color, we can see that the fragments having G1, this, these three fragments B1, B2 and B4, this is B4, they have a common region, right? So G1 must be lying here, somewhere here. Okay. Whereas G2 is present in B3 and B5. And B3 and B5, they have a common region here, this one. So G2 must be lying somewhere here. Whereas this is G3 actually. G3 is present in both B2 and B6. B2 G1, right? Here G3 was present. So here you can see that after G, this region must be, this region is common. So remove these things. This region is common. So here it must be G3. This is the position for G1, the position for G2, this is position for G3. Now we know from the haplotype, minimum haplotype region, it was A, D, right? I told you the answer that in the affected individual, homozygosity is present in between the A and D. So if this is the identified as the minimum region, so G1 is the defective gene, which we found by combining the results of this table and this table. Okay, so that was it. I urge you guys to, take a screenshot of this problem and try to solve it on your own. You will definitely follow with what I have explained. Also, sir is in his lecture, in lecture about position cloning, in that lecture, a similar problem sir also did. So this problem will be a practice problem for you guys. Okay, so today's lecture went too long and you guys are the only participants. So all the best if you guys are giving the exams, and if you have any queries, do let me know. Otherwise, I'll end the meeting.
The first 1 to 3 question, can you repeat it?
Sure. So this was the first question. While advanced genomic methods have helped us identify the genes mutated in genetic disorders, the challenge has been to dissect the mechanism by which the affected gene causes the disease. Which one of the following options offer an explanation for this challenge? Okay, so you have to identify which of these options are a challenge when people are trying to identify the gene which is the cause of a particular disease. So here the answer is D, all of these, because for most of the genes we don't know their physiological functions. Also, many of the human genes are yet to be characterized, and there are not enough types of biochemical assays available to us which can be used to evaluate the function of a gene or do functional assays. Like for example, for neural conditions, right? People have not yet able to develop very robust biochemical assays for understanding symptoms like mental retardation and etc. Okay. So this was first question. Second question, which one of the following statements distinguishes a monogenic disorder from a polygenic disorder? So if you recall what is monogenic and polygenic disorder, then you will be able to do this question. The first option is the DNA sequence variation contributes to disease onset. It can affect either sex. The affected individual can transmit the disease causing allele to the next generation. So both of these disorders, the option A, B, and C, they doesn't say that these two disorders are different. That's why option D is correct. None of these statements distinguishes monogenic disorder from polygenic disorder because A and B is possible for both types of disorders. Okay, it can affect both sexes. Genetic variation contributes to disease onset, right? If you are following, you must be agreeing with me. And lastly, it can be transmitted from one generation to next generation, right? From the affected individual to the next generation. This is the third question. Two distinct genes that are likely to be inherited together are called. So, if two genes, they are linked, they are inherited together, and two genes are always present in two different locus, like it is shown in this figure which I have drawn. Locus 1, gene A is present and its variant small a is present. Another locus B, it is present. Another gene B that is present at locus 2. Okay, so these two genes are present at two different locus, and their position is such that they are not segregated from one another, or their segregation is a rare event because of which they are called linked genes. So here the option is C. Segregation can happen, but it's a rare event. Okay, means A can be separated from B by recombination, but it happens rarely. In majority of the offsprings, A and B will be inherited together. Okay, so this is the property of linked genes. So these were the first three questions. Any other query?
Regarding the last, the order in what you have mentioned, how to
Physical order.
physical order is kind of tricky calculation which we have to do. How we can do that, because aren't any practice, I think.
So you have to remember that a particular fragment, say B1, okay, you have to maybe in first go you cannot get the correct order. Second go you will get the correct order, and third go you will get the correct order. But you have to remember that while ordering, okay, while writing the orders of the markers, you cannot write in such a way, or you cannot place the markers in such a way for the next fragments in B2 that say say S here, it cannot be after A. Why? Because if you follow B2, in B2 S was not present, right? So it has to be upstream. So you have to use this kind of logic, and it comes from simple observation. So in first observation, you will see that it is not going. So actually you can do this practice in class 12, the chapter principle of inheritance and variation. So there the linkage, how the linked genes are ordered together. Okay, so that kind of questions you can find, and for solving those questions you will have to use the same logic which is given here, the mapping, how the linked genes are, what is the physical order of the linked genes. So here also the markers, they are linked to one another, because of which like you have to link one marker to the another in the physical order. Same type of concept is applied in those kind of questions as well. So if you like to practice, you can have a look at class 12 problem. There also students study about linkage and recombination. So you can find questions by Khan Academy. So they have multiple such questions where they ask you to order, what is the correct order of the genes. Okay, they give you say A, B, C, D, okay, and then you have to order it. So that is the exactly same concept that is applicable here. Just you have to remember that how you're placing each marker, okay, and it will come maybe not in the first try, second try, third try, but you will get it. It is not that difficult. So even if anyone can make mistake in this, but it is all about observation. Okay, it is not a difficult problem. I urge you to look at this problem once again. Okay, you will get it. Okay.
So does it require any subscription like separate to look on the Khan's Academy website?
What Khan's Academy, you can look in the topic of recombination and linkage. That topic, Khan Academy. There they have similar type of questions which talk about the order of the linked genes. So here you're not ordering the linked genes, you're ordering the markers, but same concept is used. Okay.
Does it require any subscription for that?
No, Khan Academy is free, completely free. All the questions you can do for free. That's a very good thing actually for +2 students. Okay. Any other queries?
Is there any other session we gonna have after this, or it's going to be the end?
So I think this is the last week. We were told to take 4 sessions for the 4 weeks to cover the content of the 4 weeks. So you guys maybe you can talk with NPTEL people, and if they say we can be instructed to take another session, but as per my knowledge, this is the last week of discussion. Tomorrow as well, there will be few. There will be one session. Day after tomorrow, there will be one session. If you have any queries, do take that opportunity to ask them. Okay, join those sessions and ask those queries. If those queries are not coming to you today, you can ask them tomorrow and day after tomorrow. Okay. Anything else? Okay. So if not, thank you guys for joining. I am ending this meeting. You guys sure? You
Thank-
don't guys you okay, okay. No carries, no? Okay, fine.
Thank you.
I'm ending this meeting. Thank you all for joining.